Good morning.
How are you?
Good. How are you?
Good. It's been a while.
All right. I just want to talk and see if this is going to record again. I'm waiting. I think it's good. That's good. Okay. Okay. Good morning. We will get started. Any questions before I get going with the chapter? Okay. So the midterm grades were posted yesterday afternoon. There were some individuals that had completed extra credit, and that was calculated into the scores. There were some that did not, and that was not held against you. But keep in mind, if you were in the process of working on that extra credit paper, those points weren't added into your overall score. You should see all of your points on Moodle. You can divide that by 186. That's the current course total after the 2 tests. That should give you an idea of where your score is overall. So, and if you got questions or comments about that stuff, shoot me an email. Yeah. It depends on if you had it completed. If you completed it by yesterday, it would have been included in there. If you didn't have it completed, then I didn't count that in there. So it just depends. I mean, it's going to count on your score, but as far as reporting midterms, if you didn't have it done, and it's not set to close until I believe the 18th, so you do have a little bit of time with that. Okay. Okay. So I think we are good with that. Let me maybe. Okay, we left off on Monday. We got a little bit into this chapter, almost half. As we move through here, this and make sure. Now I'm getting chats. Hang on. Try to answer these questions as I see them popping up. Final score is now still out of 186. Yes, that is correct. It's still out of 186, even though, so as I'm posting things on there, they're not getting posted as specifically as extra credit. So it's posted really as an assignment. So on Moodle, it shows your course total. Definitely has more points than the 200, than the 2 exams that we have dealt with. So don't trust that number that you see. I don't know what you see on Moodle as far as point totals, but 186 is where we're at. Okay. So with that, we left off and we were talking about different types of cellular interactions. So with this image, with this figure, what we're looking at is a natural killer cell. The purple guy is a natural killer cell. And these guys have a couple of different potentials as we think about them doing surveillance and trying to recognize foreign invaders. Natural killer cells can target things as they are looking, doing cellular surveillance, if you will. They can look at our own cells and see the MHC1 complexes that we'll hit here in just a second that are on the surface of all of our nucleated cells, and they can recognize if there's issues. But they also have other possibilities, and they can utilize antibodies that have recognized foreign invaders, and those antibodies can stimulate these natural killer cells to do their job, all right, to kill, as their name should imply. So this is the ADCC is antibody-dependent cell-mediated cytotoxicity. That is a mouthful. ADCC is maybe better. So it's really just using antibodies to allow these cells, these natural killer cells, to recognize the issue. And it might be that maybe what we're looking at is a pathogen that's too big. Something, bacteria oftentimes aren't really an issue. And we could think about engulfing, phagocytizing, destroying those guys. But other things, maybe larger things that can't be readily phagocytized by some of these immune cells, need other mechanisms. So it could be, a type of protozoal cell, a yeast cell, maybe a type of fungus, or maybe a type of worm that our cells have recognized and produced antibody against. Those antibodies link up to specific epitopes on the surface, and it stimulates these natural killer cells to come in. And again, without phagocytizing, they degranulate and they dump their digestive contents and extracellularly kill these foreign invaders. And it's likely that we may see handfuls of natural killer cells all surrounding this cell, linking up to the respective antibodies, the FC regions on these antibodies. And the natural killer cell has receptors on its surface that recognize this type of immunoglobulin, the FC region, right? So this is one mechanism that aids in our defenses. And in this circumstance, it does require or rely on the fact that we've got antibody that's been produced against these in the first place, right? To do the antibody dependent. And like I said, natural killer cells, dependent upon the circumstances, natural killer cells don't necessarily need to have antibody to stimulate this in the first place. But with that said, they would be targeting our own cells that were not expressing the proper MHC1. This is some foreign invader in the first place, and it needs some way to say, hey, this is a bad guy, right? At least for those natural killers to recognize. So I mentioned the MHC1s, and this figure I think does a pretty good job of trying to describe a little bit about what's going on with this. MHC stands for major histocompatibility, and these are a series of proteins that I've described before kind of act like a molecular fingerprint. There are our mechanism that our cells express to say, this is me. And there are other cells, immune cells, that have another group of histocompatibility complexes that allow them to do surveillance and communication with one another with these types of proteins. So all in our bodies, all of our nucleated cells have MHC class 1 molecules on them, right? And this is important. This is our molecular fingerprint, all right? There are, the erythrocytes, your red blood cells. Those cells don't have a nucleus. They are cells that lack this complex. They don't have MHC proteins on their surface. That becomes important when we think about the possibilities for blood transfusions and the ability to give blood from one individual to the next. There's definitely issues as far as compatibilities, and you have to have a specific type and receive that respective type. We'll hit more details with that and transfusion reactions in the next chapter. I don't want to go off on that right now. But the red cells don't have this MHC1, and that in turn allows for people to donate blood. So a type A person receiving type A blood from somebody else, all fine and good, all right? And like I said, I'm not going into compatibilities right now because there's definitely some other potentials that can be going on with that. But that's the reason for that. When we think about organ transplants, somebody needing a liver or a kidney or other stuff, whatever, all right, this becomes an issue. And the MHC class 1s are definitely things that we consider. Bone marrow transplant is maybe a good example with that. And they try to see what your complex is, what is your protein profile, if you will, and they try to find, when they're looking for organ donation, they look for the closest match as far as these complexes go, all right? And you're not going to find, unless we're talking an identical twin, you're not going to find a perfect match with these proteins, but you can find something that's pretty similar, meaning the potential to recognize that as being really foreign is not as much. And so that minimizes the risks of rejection when you get as close to a match as you can, right? Uh, and that's important. I think I've mentioned this in a previous lecture. If our red cells had these MHC1s, you couldn't just expect to get a blood transfusion from anybody without having as close to a perfect match as possible. And that would be big issues. All right. So all of our nucleated cells have this class 1 molecule, and then some, not all, but some of our immune cells have this extra suite of proteins that allows them to communicate with one another and basically show some of these bad guys to other cells. So macrophages, dendritic cells, and B cells have these class 2 molecules. They also have the class 1. They've got both, all right? But this is important when we think about cellular communication, really talking about the bad guys that we've possibly come in contact with. So this suite of molecules, this MHC class 2, is important when we think about this communication. And back to those natural killer cells that we were just looking at, if we were to have a cell that was infected, like a viral infection, or possibly cancerous cells, not all, but some of these cancerous cells, the class 1 molecules end up being altered or potentially non-existent. And those natural killer cells, as they're doing surveillance, recognize this problem, this defect with our own class 1 cells, and it basically stimulates them to target and kill those cells, thinking, the cell's infected with virus, we should destroy it, as an example, because this class 1 molecule would be very different, or like I said, potentially non-existent. So this image is kind of showing what's happening with this process of recognizing the bad guys in the first place. So dendritic cell, like kind of macrophage-like within your skin, is really what we think about with these guys. So we start off here with a couple of different bacteria that are going to be recognized by this dendritic cell. So it gets phagocytized, it gets engulfed as the cell recognizes and pulls it in. It brings it in, and it's enclosed within a vesicle, a phagocytome. So it's membrane bound inside of the dendritic cell. As it gets in there, lysosomes fuse to this phagocytome. That creates a phagolysosome, or a secondary vacuole. But ultimately, the idea would be all of the digestive enzymes, all of these substances that are going to help aid in the destruction of this foreign invader. The lysosome dumps it into this phagosome, and now we kill these bacteria on the inside. In the process of destroying them, there are fragments of the bacterium that are available. And we mentioned some of this stuff before, these antigens, oftentimes protein molecules. There can be some other mixtures within there, but protein substances, and within these antigens, specific regions, those epitopes that we can recognize as being different, right? That's what gets packaged up and presented on the outside. And it's presented on that MHC1, all right? On that complex or that suite of proteins, it shows these little epitopes. And now this cell is basically, it's kind of show and tell like. It's now trying to tell all of the other cells around it, look at this bad guy that I have found. Here's pieces of him. It's like, it's like a mugshot, trying to show the bad guy so that all of the other cells around are like, oh, I see this. I can go out and search for more of them. So now they're all looking, and you could think of these little red blobs on the outside of this bacterium. These are the specific epitopes signatures that this bacterium is bad news. And these other cells that have now been stimulated by this dendritic cell to seek out these cells, basically kick it into overdrive and try to ensure that the bacteria or whatever foreign invader doesn't have the opportunity to cause serious illness. It's what you would hope. Obviously, the next unit of material, we're going to be talking about disease, and there's definitely mechanisms where these organisms have the potential to get around that. So this image is an electron micrograph of a T lymphocyte. And this T lymphocytes are huge players in an immune response. It says here it's responsible for cell-mediated immune response, and yes, that is true. But really, they help to govern all of the immune responses that we think about occurring inside of our body. T cells play a huge role in ensuring that we mount adequate responses in the first place. And there are different suites or different classes of T cells, and their roles or functions are definitely varied. But we do think of these guys participating and orchestrating this response more so than just cell mediated. And as I mentioned before, when we think about this stuff, these types of responses, cell mediated or the antibody or humoral mediated response, we talk about them as separate occurrences, but really they work together, right? And it's these T cells that help to orchestrate that. We showed some images of this, these lymphocytes before when they're stained up, and you really can't tell the difference between a T lymphocyte and a B lymphocyte that we'll see here in a bit under a microscope. They look like just a large nucleated cell with the bulk of it being nucleus and very little cytoplasm available to be seen in that cell. Pretty good indication you're looking at a lymphocyte. This, again, we're looking at an electron micrograph. We're not looking at it all stained up. So it depends as this next two figures really go. It depends on the types of cells as far as where we might see them maturing or originating. The B cells within the bone marrow and T cells maturing within the thymus when we're thinking people, all right? So bone marrow we could see in the head of the femur, also up within the scapula. There's areas with marrow, typically thinking stuff like bone marrow transplant. Usually it's the femur or maybe the ilium. They tap into one of these areas where they try to extract marrow, and usually right up within here. So there's all sorts of possibilities within the marrow for more than just our B cells. We see the hematopoietic stem cells that originate with all of these, and then we see maturation. As I said, the B cells maturing within the bone marrow, T cells migrating and maturing within the thymus. So that's what we've got going on here, and you don't need to get bogged down with that stuff. The T cells, like I said, playing a huge role in governing all of these different types of reactions. So there's a receptor on the T cell that allows these cells, these T cells, to recognize and communicate, if you will, with other cells. So this is the plasma membrane, and then here we've got an antigen binding site on this T cell that allows it, again, to recognize and communicate. So it is able to come in contact with cells and recognize MHC1s or MHC2s or potentially both. right? And like I said a second ago, there's a handful of different types of T cells, right? We mentioned those natural killer cells. There are other cells that are very similar to natural killer cells, cytotoxic T cells that can recognize these foreign cells based off of their MHC, or not foreign, our own cells, but recognize that there's something that appears foreign about them and cause their destruction. Others would think about recognizing those either dendritic cells or macrophages, something that's presenting those antigens. It presents it to a T cell, which in turn allows it to recognize the specific epitope and really to differentiate and to decide which classes of cells might need to be tasked to deal with this. Should we go the cell-mediated route? Should we target B cells and stimulate them to produce antibody? Maybe both. So here we've got this activation, as I just basically kind of described, and the possibility for things to occur, all right? Just depends. So we start off with a type of T cell called a T helper cell. This T helper cell has the potential to differentiate and stimulate different cells to recognize, all right? And it depends on what's being shown in the first place, all right? So here we've got a dendritic cell. This is an antigen-presenting cell. It's back, a couple figures back where we've engulfed that bacterium, we've destroyed it, and now we're presenting bits of it on the outside. As we present those bits, that's when we are this antigen-presenting cell. So little epitopes are expressed or exposed in this MHC2 molecule that basically have the ability to stimulate these T cells, and as they look at it, they differentiate accordingly, all right? So at this point, it's a naive helper cell. It has yet to decide what it should do, all right? Here we've got the dendritic cell. This is just a blow up of the dendritic cell and the T helper cell coming together, all right? So, and I don't remember if there's another blow up of this or not, but we'll back it up to this. This on the T cell and right here on our dendritic cell are interacting together, all right? These two molecules come together right here and are able to exchange information, if you will, all right? So this naive cell gets activated. It gets told, here's this bad guy, or this fragment of a bad guy, all right? And you should recall that on an antigen, typically there are multiple epitopes. There are multiple pieces on that antigen that can stimulate a response, all right? So this one bad guy that gets phagocytized and destroyed may have a dozen different signatures, maybe even more than that, that it's, stimulates the potential response within us, all right? So this dendritic cell, as it's presenting antigen, could have multiple things that it's presenting, all right? And it could be presenting to multiple T cells simultaneously, showing all of the bad bits to numerous types of cells. And this is where we see kind of this concerted effort to stimulate as many arms of the response as you possibly could, all right? So here we're just showing one, all right? So this one epitope is being expressed on this class 2 molecule, and it shows it to initially this naive cell. As it shows this, as they link up and basically exchange this information, this T cell becomes activated and the epitope gets passed from the dendritic cell to the T cell. What we do after that depends on what was shown, all right? So there's multiple fates. We could go to the T helper 1 route, T helper 2 route, And there's also likely in either of these circumstances, you see some of these cells that are memory cells laying in wait for a subsequent encounter with this, right? T cells and B cells both produce memory cells, right? And that helps with subsequent reactions or infections for that matter, right? So it depends. And there is a table. I don't know if I need to toggle over to the table or not. It's not in this PowerPoint. But if we stimulate the T helper 1 cells, that's basically telling the cell-mediated response, we need to kick it into gear. So we think about things like stimulating more macrophages, more of the neutrophils and eosinophils and basophils potentially that can come in and respond. These types of cells get targeted by T helper 1s. And the T helper 1s release a suite of cytokines, chemical messengers that help to call in those specific troops. Depending, this epitope might be a little bit different, and it's something that could stimulate these naive cells to differentiate into T helper 2s. The T helper 2s turn on a different set of cytokines. They start releasing different chemical messengers that tell the B cells, you ought to come over here and see what I found. These B cells will link up to these T helper 2 cells, and the T cell now shows the B cell what the dendritic cell showed it in the first place. And once it shows that, that B cell sees it, sees that epitope and says, oh, I could make something for this. And it starts producing antibodies that are specific for that epitope. And again, potentially multiple epitopes being presented. So now you got the possibility for numerous types of antibodies, all against the same bad guy, to be produced basically simultaneously, all right? And like I said, maybe different epitopes have stimulated both the T helper 1s and the T helper 2s, so you get both classes coming in and trying to respond. And really, in an immune reaction, this is what you see happening. Usually both players, right? Not always, there are some exceptions, right? And again, some of these will basically get directed to become these memory cells to kind of lay in wait. And once this foreign invader is back again, they kick this situation into action much more quickly. Okay, so here we've got more with this with our, this is a cytotoxic T cell. So this is one of the different types of T cells. The T helpers, we typically call these guys CD4 cells. And there's CD4s that are helper 1s or helper 2s. These cytotoxic T cells, we call them CD8s. And the CD for cluster of differentiation doesn't matter. You don't need to worry about that. It's just a different group of proteins that are activated within this respective type of cell. All right. So here you can see it up here, a CD8. And on this other one, CD4. All right. So CD4 allowing recognition this way, CD8 we're still recognizing, but it's a different type of cell. All right. So this unactivated cytotoxic T cell sees what's being presented, and in this case, this is our own cell. All right. So we've got an intracellular pathogen. So one of our own body cells gets infected, potentially with a virus. And in this case, it doesn't have to be a virus, but it's infected, and the MHC1, which is our own molecular fingerprint, it now shows something's up. All right. It presents something in there, basically a flag raised saying, we've got problems, we've got trouble. right? These guys, these naive cytotoxic T cells, are doing surveillance, looking at our own MHC1s, and if they come in contact with an MHC1 that's got something exposed in it, it stops and mounts a response. It becomes activated, right? And as they dock and link up together, it stimulates, as it sees this, stimulates the cell to respond. As it becomes activated, it then starts searching out these cells, linking up, and basically destroying them extracellularly as it releases all sorts of different enzymes. Again, from these lysosomes, all sorts of different molecules that are destructive. Perforin that will poke holes in the plasma membrane, and granzymes that are all sorts of different types of digestive enzymes that target the destruction of these cells, right? So stimulating controlled death or apoptosis, where this cell basically takes one for the team to hopefully prevent other cells from becoming infected as the pathogen replicates within it. So another class, and this would be part of the really thinking kind of cell-mediated response as we move down that way. Okay, this next one, our macrophage. The macrophage, again, a great player when we think about its potential to phagocytize cells. As it does this, it becomes an antigen-presenting cell, just like that dendritic cell that we started with thinking about it. So the macrophage up here, as it engulfs, and we're only showing the class 2. It's got class 1 molecules on it as well. It is a nucleated cell, all right? But on this class 2, as we see, and we've already phagocytized, we've pulled it in, we've engulfed it, we've destroyed it, and now we're presenting these fragments. We're not showing any of that stuff, all right? That's already happened, all right? We've presented this little fragment, this epitope on the class 2 molecule. The T helper cell sees this, right? And you can see the CD4 molecule right here and the receptor. These come together. The CD4 basically helps to facilitate the docking, holding them together as they link up, all right? So this happens, and we come in and potentially stimulate. In this case, this epitope is not recognized, right, by this receptor, right? Or so there's multiple potentials with it. So we can recognize it, go one or two routes, not recognize and not stimulate a response. And there's other potentials here where we see this super antigen that can cause like an overstimulation. So the super antigen, it bridges this gap without needing that respective epitope, and it's an uncontrolled activation. This is something that oftentimes it's been described as something like a cytokine storm, and this is definitely something that potentially can be happening inside of your lungs with COVID, where you can see these specific interactions causing an overstimulation. You think, releasing cytokines, these chemical messengers to call in the troops, could be good and usually is, but an overstimulation is not, and it definitely can cause serious ill effects, potentially massive inflammation, and, well, definitely problems, all right? So just depends on what ultimately is happening with these recognitions. Perfect situation, we come in, and again, dependent upon that epitope, stimulating this naive T cell, either to differentiate into a T helper 1 or into a T helper 2, all right? The CD4s, they're either helper 1s or helper 2s, all right? So that's kind of the idea. Okay, so B cells, shifting it over to these guys, there are all sorts of different potentials when we think about this. They're the same as we think about the potentials with those T cells and the potential for them to recognize and stimulate responses. It's tremendous as far as what their specific binding sites really look like, if you will. All right. So B cells, and as I said before, looking at a B or a T cell, you can't tell those guys apart if you were looking at them on a blood smear. All right. The B cells have receptors on them, and these receptors look very similar to an antibody that we've already kind of shown. And this may be like that IgD antibody that is important with interactions on B cells. So this allows recognition of specific foreign substance with their receptors. So here we can see a couple of interactions. Oops. So this is a T cell independent activation. So our B cell with its specific B cell receptors can recognize some things without needing stimulation from a T cell, all right? So these receptors can recognize some fragments on bacterial cells, in this case polysaccharide antigen, in this case repeating fragments of that these receptors recognize and can link up. Over here, we've got antibody, IgM, the pentameric immunoglobulin, that's being released as a result of this recognition. The B cell sees it and stimulates the production of antibody. All right? And in this case, like we said, doesn't need a T cell to show what's going on. All right. So there's there's receptors. The PAMPs are the pathogen fingerprints, if you will, that allow this interaction to really see it as being formed. We don't need to worry about that. All right. So B cells can be stimulated independently if these proper receptors can recognize, but oftentimes we think about T cell dependent activation, where it's already been shown something that, like a macrophage or whoever has destroyed, it stimulates accordingly. All right. And that's kind of the same. Well, here's an idea. So the T cell dependent, you can have, in this case, the B cell that's making this initial recognition. It could be a macrophage or a dendritic cell, either way, all right? So this case, we recognize this bacterium, we internalize, we destroy, and can present. And that's really what's happening right here is antigen presentation, epitopes being expressed. And that shows this T cell, the bad guy, if you will, that fragment of it. As this links up, that T cell sees it, and you see all of the little granules, the cytokines start being released. These cytokines are calling other cells. And again, it depends on that type of epitope. So with what we're looking at, this T cell sees this epitope and it stimulates B cells to come in and respond. So this T cell differentiates and becomes a T helper 2 cell. And the cytokines that are released stimulate B cells to show up. As these B cells show up, you get more of this interaction where the CD4 molecule shows these B cells and tells them this is what we need to be looking at. And again, potentially multiple epitopes. You think about these interactions, you don't just make one class of antibody against one single piece. There's numerous antibodies that are being generated against as many bits of that organism as you possibly can. All right, I kind of feel like, the more ways you can target it, the better. All right, so each one of the respective B cells, however, has specificity. So it's only making that one type of antibody, right? So if you produce antibodies against 10 different epitopes, there's 10 different classes of B cells that are making these things, right? Pumping them out. There are two fates with the B cells as well. Some of them become memory cells, which are important when we think about protective immunity, same as the T cells producing some memory cells. But the bulk of them become plasma cells. Basically, when that B cell gets stimulated and starts producing antibody, it becomes a factory, really, where it's secreting these proteins, and that we give them the new term, a plasma cell. They're still B cells, but they are proliferating or pumping out antibody, right? And there's all sorts of potentials with that, as we kind of showed in the figure with the primary and secondary response. Some antigens stimulate a very good response and you get a lot of antibody produced, and some are not so good, and the antibody levels are very kind of low, kind of marginal, right? And so levels of immunity might definitely be different as well. Thinking about vaccinations, you might have something that requires one shot and you're good to go. And oftentimes booster shots to truly get those levels of immunity up there and keep it prolonged. After that, do two shots, your primary and secondary shot. Does that confer lifelong immunity? Depends on the specific vaccine, truly. All right. Okay. So this is, again, B cells as they start proliferating and generating those specific immunoglobulins, those molecules. So this image shows both. It's kind of like what we showed Monday with the primary and secondary response, but it's got both classes. IgM, this immunoglobulin, again, at the pentamer, the 5 linked together, 10 binding sites. It's the first to be produced. And then IgG takes over, all right? So here in this figure, we've got antibody concentration on the y-axis, we've got time on the x-axis, and we've got a little jig-jag in here in the middle, where we've got, potentially maybe months in between. It's hard to say. Matters not. So the primary exposure, it takes time. It takes maybe 2 weeks to start getting measurable amounts of antibody, primary antibody, because it's your first response. So the first bit that starts being produced are the IgM molecules. As they start to decline, the second class of immunoglobulins being generated, the IgG. It shows up about 3 weeks after that primary exposure, peaking at about that point, and its concentration is more than the IgG. Both of these decline and go back to nearly baseline levels, all right, to almost maybe being undetectable after the course of, a month or two. That level might go down to unmeasurable levels. You kind of hope that that's not the case, right? So again, a specific amount of time passes. There's a secondary exposure. This could be a booster shot, or this could be coming in contact with the real player, whatever it is, all right? Now, as you look at this, we've shifted the time considerably. IgM kicks in, and we start producing it within just a couple of days after this exposure. We've got measurable amounts, what took almost 2 weeks. And now, within about a week's time, we've got our peak of IgM. IgG the whole time is increasing, and those levels are considerably more pronounced. Again, if it's a good epitope, if it's a good antigen. And they stay prolonged much longer than you see with that initial primary response. Leveling off a little bit, but hopefully persisting longer. And we think about possibilities again with memory cells. Keeping these levels elevated are really the results of booster shots to keep these levels higher than we see over in this circumstance, right? So this could be, again, a vaccination situation. It could be coming in contact with foreign invaders, all sorts of possibilities. We could, although we're not showing them up here, we could think about an allergic reaction, and we could show IgE on this figure as well. And there is the potential for these immunoglobulins. There can be some class switching. We might be producing one type and then shift it over and produce another. We're not going to get bogged down and go into details with that stuff. But similarly, you could see with this primary response that not only are we making this, but we also maybe make a little hump of IgE. The IgE is that immunoglobulin that's important when we think about allergic reactions, right? So you'd have that initial sensitizing dose, you'd get a little bit of IgE, and then you would get a shocking dose, that second exposure, and that IgE would spike and it would stimulate immune cells like mast cells to degranulate and dump lots of histamine that in turn stimulates a lot of your ill effects when you think about allergic reaction. So I think we are very close to the end of this. I'm almost out of time. So I'll just wrap this up. The mechanisms of getting immunity, it depends on the circumstances. They can be naturally acquired or artificially acquired, and they can be passive or active, right? So naturally acquired, it's immunity that you get passed from mother to offspring, crossing the placenta, IgG does this, or through breast milk, right? So it's mother's immunity passed to the baby. That's natural passive. Artificial passive would be getting plasma from someone else. That plasma harbors antibodies. You're picking up their antibodies, right? It's not long-lived, right? In that circumstance, artificial, somebody else produced it, and you got it passively. The active immunity could be through illness and recovery. So you've got sick and recovered. That's a natural active, or the artificial active would be vaccination. So you've been given something, often killed strains that stimulates the immunity like inactive, but it's been created differently. So real quickly, variolation for smallpox, this was immunizing, if you will. There were all sorts of ideas with this, picking scabs, grinding them up, and blowing them up the nostril of someone else was the first thought of helping to prevent infection, variolation. But we often saw individuals getting sick. So Jenner was one that came up with vaccination for vaccinia, cowpox, and he noticed the cowpox lesions on milkmaids. If they were infected with cowpox, which is a close cousin, if you will, to smallpox, these people couldn't become infected with smallpox because there was kind of a cross reaction with this immunity. And he in turn used the cowpox, which was much less virulent, as an immunizing agent, vaccine, that helped to really start this whole process of vaccination for all sorts of things. But smallpox was this initial. And there's all sorts of historical stuff going back to that. I'm not going to get into it right now, but there is some interesting stuff with Jenner and his research. I think if he were a scientist today, he'd probably get thrown in jail. All right. Questions or concerns? Yes, sir. Yeah. Maybe. To this guy? On the second bit. So IgM is, that immunoglobulin is something that's produced pretty rapidly, but it's not something that maintains. You don't see a lot of memory cells with the IgM. IgG produces considerable amounts of memory cells. IgM not as much, and so you see the level start pretty quickly, but it drops off. So the question, why IgM dropping off like this, and it's really not as many memory cells produced with this. So is that good? Any other questions? Let me check this. No chats. Okay, guys, have a great day. We'll see you Friday. We'll start with chapter 19. We should be able to wrap that up and have some time for some review as well. And there is a study guide posted on Moodle if you have not seen it.